Yes, okay. So to start off, we just want to really just go over research, what it is, and what it means. So in a sense, we all have used the word research colloquially, right? We all know that research is the act of finding information, is the act of learning something about a system or just to answer a question, right? Like when you apply to colleges, you research the different colleges, you learn more about them. In science, though, the term research has a more specific meaning. Usually it's to answer like a very specific scientific question or to learn more about a scientific process. And to do so, historically, what you might have been exposed to is the scientific method. And what that basically says is, okay, we have an observation, right? A hypothesis that you make to explain that observation, and then you test that hypothesis with an experiment, collect data, analyze it, and then communicate your findings, right? And this is something that you've probably seen a million times before, right? Something you've seen in high school, maybe even in middle school. And so you might be familiar with this model of research. What we're here to show you is a different kind of model. So not all research has to follow the scientific method, right? Research is more widespread than just hypothesis testing. And while there have been many, many important advancements that have been made with that kind of testing, there are different types of analyses and different methods you can follow and different approaches you can use to make meaningful conclusions and to just learn more about a system, to be curious and exercise and explore your own curiosities. So the two biggest ways that we're going to be talking about in this call are literature reviews and exploratory studies. So what those two are, literature reviews are essentially synthesizing information from a number of articles and usually just using that to explore a topic. And exploratory studies are basically a type of study that doesn't really involve having a hypothesis in advance. You're kind of just exploring data, looking for trends, and just looking to get more information out of it, right? So in both of these cases, you don't always have like a clear-cut hypothesis. You're not always testing something with an experiment. You're just looking at what more you can understand about a system, and you're just doing research at the more basal level, like what is research fundamentally. And so I will hand this off to Jessica, who will talk about the normal paths to research.
Yes. So I'm Jessica. I'm part of Cabinet. So I'll talk about the normal path to research and mentorship in research. So the three main traditional paths to research are the high school, college in-class, and cold email routes. So for the high school route, there are many people who have been doing research since their junior or senior year of high school. Some high schools have programs just for research. High school students can also apply to research labs outside of their schools. I know a few people who applied to do research at schools like NYU, but not having research experience in high school doesn't necessarily put you at a disadvantage in college. I know I didn't have research experience when I applied to the lab that I'm currently in. So you just have to show them that you're willing to learn when you get accepted into lab. So for the college in-class route, you could take a research class. So here at Stony, the 400 level bio courses are where you have to complete a research project under a faculty advisor. Another way is to go to your professor's office hours. And I know that most professors here do some kind of research. So going to their office hours is a good way to get to know them and ask them more about their lab.
No, the shot they gave me.
And lastly, we have the cold email route. So basically, you would find 10 to 20 professors whose research that you find interesting, and you can shoot them an email. Uh, what I did was that I made an Excel sheet and listed the labs that I was interested in. So in my Excel sheet, I put the name of the PI, their email, their fields of research, so like psychology or neuroscience, etc., and a paper from their lab that I found interesting. So I would definitely personalize your email because each lab's work is different. And I would put a paper that the lab has published. I would read it, and I would talk about why you found it interesting in your email. And you should also attach your resume or your transcript at the end of your email before you send it. So next, I'm going to talk about mentored research. So research can definitely be overwhelming, especially when you first join a lab. There's going to be so many lab techniques that you'll have to learn if you're doing wet lab research. You'll also have to read research papers and learn how to follow protocols for designing experiments. But if you have a good mentor, then they'll help you understand things along the way. mentors are extremely knowledgeable in their field and they have years of experience. And the skills that you build under them are transferable to whatever your future career may be. And your mentor doesn't necessarily have to be your PI. So in my lab, I work very closely with the PhD student and a master's student. And whenever I have questions, I would always go to them.
So now that you know about mentor research, right? You might be super excited, and you might have already tried some of these things. I know a couple of you might have already tried emailing professors. They're trying to get into their labs, and we've all run into this issue that's kind of pervasive and affecting our entire country and the way that we live our lives, which is the pandemic. So a lot of you are finding it nearly impossible to find mentors now. A lot of mentors are not, they don't really have space in their labs because of restrictions with social distancing. So the pandemic has really reduced in-person lab capacity. So a lot of PIs, first of all, they might only be allowed to have one or two people in the lab at a given time, right? So they can't have someone that's inexperienced working. Because they need to train them, right? That's one really cool part of research, that you get trained to use a very specific skill set. And the other issue is with social distancing. If you need to be 6 feet apart, you can't have a PI right next to you showing you how to use different equipment, how to pipette, how to run a gel. And so because of the pandemic, there have been a lot of restrictions on the lab capacity that, different professors are able to accommodate for when they get new students into their lab. And we can say that lab positions will open up when the pandemic is over, right? But a lot of you are here because you want something to do in that meantime, right? You're at home, you want to be productive, you want to be involved in the research process. And maybe you could work on skills that will help you get into a lab in the future once those lab positions do open up. And so what we're really going to be addressing is how you can work on skills that are necessary for research and make yourself more appealing to a lab once the time comes, right? So we're really going to be focusing on independent research you can kind of do on your own without a mentor for the meantime. So what is independent research? Independent research, by definition, is research that's independent, right? It's work that you do primarily on your own. So you are the mastermind. Think of it as you being your own PI. You're the one that's choosing the topic, designing the study, and formulating your own conclusions. And though, historically you do this with a mentor, right? You can do a modified form of independent research where instead of, creating the next publication that will be published in Science or Nature, right? You can focus on skill development and building familiarity with the research process. And maybe you can find something that's cool and exciting, but the real focus is on just getting yourself ready for the future and preparing skills that will just help you in the long term. So what can you do, especially from home, right? A lot of you are obviously working from home, and all of this presentation is really going to be focused on methods and techniques that you can really work on from home and are still applicable to your research and to research later on if you choose to pursue that. And so we also wanted to really discuss things that are affordable as well. So we're going to be focusing on, as mentioned earlier, literature reviews and also exploratory studies. So literature reviews, as I explained earlier, they're essentially just a form of research in which you synthesize information from a number of articles to explain a topic or to answer a question, right? So one question you can try to ask is, what's the role of P53 in cancer, right? P53 is very well documented, and so is cancer, and its role in cancer is especially well documented. So if you're seeking to develop your skills in research, to practice reading articles, because that's the one thing that any researcher is going to spend most of their time doing, just like reading articles, just practice by trying to form a fundamental theoretical understanding of one question or one topic. And this is really useful because not only are you learning how to read scientific literature, right? But you're learning how to find relevant information from research articles. You're not just like summarizing one article, which is something that many of you guys are probably doing for classes like Bio 202, right? You have to like summarize an article. You're not just summarizing an article here, you're finding relevant information and kind of piecing it together, telling a story, and just building a foundation in an area of science. And what's really cool about this is actually, if you decide to write a literature review for yourself, what you can do is you can target it towards your interests into labs you want to apply to for the future. So let's say you're really interested in cancer, right? And there's a lot of cancer biology labs on campus. You can just do this literature review in your free time, take initiative to do this kind of activity, this kind of exercise. And when you apply to labs, you can just show them like, hey, I've been working on a literature review to understand your topic better. And the fact that you're willing to understand that field even more is something that is really, really impressive from a PI perspective. Another thing that we're going to be talking about, which I'm personally super excited to talk to you about, is exploratory research. So like, what's exploratory research? Like I said earlier, it's just the process of exploring data. You don't always have like a specific hypothesis in mind, but you're essentially just looking for trends. They might exist, they might not exist, but either way, you're just practicing statistical methods and really just kind of making findings, which is cool. You're not really running experiments, but you're making conclusions on data, which is really, really, really cool. So from home, right, obviously you're not going to be able to whip out a colony of bacteria and start conducting tests, but what you can do is you can take advantage of public databases that exist out there. So there's so many of them. There's census data, there's the Department of Bureau and Labor Statistics data. So these are like, and this, so these two I would say are very like sociological data that you can use for like demographic analyses and demographic research. The CDC data catalog is something that's super, super cool. And it's really cool for those of you that are interested in like public health or just health medicine in general. Basically, it's just a database of a lot of public research that's basically going on. It's a lot of Excel files and CSVs. And there's a lot of COVID data in the CDC data catalog. So if you want to do like an independent project, just exploring and learning more about COVID, the CDC data catalog is a really, really good option. And finally, my favorite one, although I guess I'm biased, is the NCBI Bioinformatics data set. So it's basically a collection of all the nucleotide-based data sets that are out there. And so first, I just wanted to go over some of the more like sociological and demographic data sets and how you would approach them, right? So how do you start working with those kind of data sets, like with the CDC data set or like demographic data sets, right? So I would recommend to try to just practice doing some statistical exercises, the kind of stuff that you might have done in any entry-level statistics course, right? So like, for example, asking a simple question, does someone's age correlate with the chance of them getting COVID-19, right? So I don't have like perfect access to the chat right now. I'll like try to figure out how to make it work, but does anyone want to guess like what kind of statistical analysis or statistical method you could do to see if someone's age correlates with the chance of them getting COVID-19? You can also like unmute yourself if you want. Cool, cool. Okay, I'm getting some DMs, so I'll just like give you guys the answer then if you guys are all shy. So basically you would just do a linear regression, right? Like I'm just gonna zoom ahead, but like basically like plotting an X and Y axis and just seeing the correlation between the two variables. Something that you can easily really do in Excel. And if you want, you can learn how to do it in a statistical analysis language like R, because RStudio is free and publicly available for anyone to download. But it's a really cool thing. You can just practice doing in your free time and just show some initiative in learning how to practice research and do statistical methods. Another thing you could try to ask is, do certain groups of states have higher COVID-19 prevalence rates, right? Technically, I guess you guys have already seen the answer, but for those of you that might not have seen the answer, does anyone want to guess like what kind of approach you would use to answer that kind of question, or what kind of statistical analysis you would do? Awesome, awesome. Okay, so I am getting DMs. So basically, yes, it is a t-test. So it's just like, you know, comparing means, and it's something that's very simple, very straightforward. It's something that doesn't really take, you know, a complex algorithm or formula to learn how to do. It's something that is easily done in Excel, easily done in other languages if you want to learn how to do them, like coding languages. But working with data sets is a really powerful way to practice just working with data and understanding how to work with it and how to perform kind of like research in the sense, because once you answer these questions, right, you have like a trend that you can talk about. You can maybe say, okay, we know that statistically someone's age positively correlates with the chance of them getting COVID-19, right? So you can like write a mini paper and be like, okay, so knowing that, right, what might explain that? What papers are out there that can explain like age and COVID-19 having a relationship? So you can really just like do things that are powerful, even if they've already been done before, right? You might find literature where someone has already found a correlation between age and COVID-19. But the act of you doing this and taking this initiative is something that's super powerful and can prepare you for the future, really. And the other method that I really, really love is bioinformatics. And so bioinformatics quite literally is the study of biological data, right? Like big biological data, aka DNA. So what do we know about DNA? Does anyone have any like facts they want to put in the chat about DNA? Anything you know about DNA? It can be the most fundamental stuff that you know about DNA. Yes. Awesome, Jessica. Anything else about DNA? Yes, there are 4 bases, which are fundamental. And that's really part of the core of bioinformatics, being able to analyze like all the different letters and just take advantage of the fact that DNA is extremely straightforward and easily analyzable. Anything else? What does DNA code for? Yes. So basically, DNA codes for RNA, which codes for protein, right? And proteins are the doers in your cell. They do a lot of the enzymatic activity for the cell. So if you can analyze DNA, right, you can really analyze life, which is a really, really cool fundamental concept. And bioinformatics is a really cool and publicly accessible way of doing that. So we know that all living organisms, right, because we don't consider viruses to be living organisms by other definitions, they all have DNA, right? And DNA codes for RNA, which codes for proteins. Genes that have similar sequences, right, are going to code for similar RNA, which are going to code for similar proteins. So that means similar genes, similar sequences are going to have similar protein functions, right? We can really take advantage of that. And there are public databases on genomes and on gene sequences, right? So you don't need like a password, you don't need $2,000, you don't need a stipend. You can access all of life's DNA that has been found on public databases, which is really, really cool. So what can you do with bioinformatics? And this is kind of like work. Oh, I realized that I paused screen share. Yes, so this is the slide. You probably were like, what is she doing? But yes, there are public databases on genomes and gene sequences. So what can I do with bioinformatics? I wanted to run through some examples with you guys, right? Just so you can really see how simple and straightforward the process is and just like the amount of potential that's out there, right? So you can use public analysis tools and compare genes, right? So one really popular one is BLAST. BLAST is basically like the Google of DNA. It'll take a query sequence, right? So kind of like your Google search. It'll take a query gene or a sequence or protein, and it'll search for similar genes or protein sequences or whatever you're looking for. So you can use this to answer questions like, okay, what genes are similar to the COVID-19 envelope protein? So what I'm going to do is I'm going to just like drop this thing in the chat. Or actually, you guys can follow along. I think that'll probably be easier. You just search like NCBI, and then you're just like gene database, right? The very first result is going to be the NCBI gene database, and you can also follow along with the recording for those of you that are like, oh, she's going too fast. We're going to just search for COVID-19 envelope protein. My laptop is fairly slow. I apologize. I'm on Tubman Wi-Fi, so this will just take a hot second. Okay. And I can just click on this, which is for the envelope protein for coronavirus, SARS coronavirus 2. And basically, this page is really cool because it just gives us a snapshot information about everything that we know about this gene, right? So like a summary about this gene, what chromosome it's on, where it's on in the chromosome. So it's easy to get lost in it, but we have a very specific goal. We want to run BLAST on the protein sequence. So what I'm going to do is first, I'm going to go on the protein page for this, right? So I'm going to hit protein over here. I'm just going to grab the first protein sequence, and I'm just going to run a BLAST. And I know it kind of went fast, so that's why like we have the recording, but basically we're on this blast page, right? And I'm just gonna hit blast, go for it. It'll take like a minute or two. But does anyone kind of have any questions in the meantime about like blast and what it does? Nope. Okay, so we can just like kind of watch this page load. It probably won't take too long. Especially since I ran this earlier. So it should be fine. So basically we get a lot of data. You can easily download this too and like or put it on Excel, whatever you want to do. But what we see is this kind of like summary sheet, right? And it gives us a lot of information, but the first thing we can see is that the results that come up are different versions of the COVID-19 genome and the different COVID-19 envelope twos, right? And we kind of expect that because there's been a lot of analyses on COVID-19. But what's really interesting is that they're not all identical, right? The percent identity is not the same. So they're not like identical everywhere. So you can honestly like, first of all, analyze the percent identity differences, like see how conserved or how similar different COVID-19 genes are if you wanted to. But what I want to do is I want to see other species, right? So I want to exclude the SARS coronavirus 2. So I'm going to just take out SARS-CoV-2, and I want to see like, okay, outside of COVID-19, like what's similar to the envelope protein, right? And we can see that it's really just similar viruses. So the envelope protein for the bat COVID, for the bat SARS like coronavirus is one hit, as is just the original SARS envelope protein. So that's just interesting. We could really like repeat this kind of analysis for any kind of gene. It can be P53, it can be FOXP2, it can be any of those famous genes that you guys have heard of, Sonic the Hedgehog, or it can be more niche genes that you may not have heard of, such as the metalloid 1A protein, for instance. It's really like, you know, the world is your oyster in a sense. Another kind of question that you can, another kind of thing you can do with BLAST is search for align similar genes and see how similar they are. So I guess it seems a little bit redundant, but I think learning through example is the best way to learn. So we're just going to go back on BLAST, go back on the gene database, right? Go here. We're going to just search for FOXP2, and then I'm just going to search for human. And actually, I'm going to just jump right ahead and just look at the proteins, right? And we see here that we have the human FOXP2. I'm just going to open this in a new tab. And what I want to do is I want to see how similar human FOXP2 is compared to mouse FOXP2, right? So I'm going to do the same old, you know, running BLAST, right? So I'm just going to, actually, I'm going to go on this version that actually takes me to the protein page. I'm going to hit run BLAST. But this time, what I want to do is I want to align two or more sequences, right? And I'm just going to type in mouse. And we'll just grab the mouse FOXP2. And basically over here, they ask for an accession number. So I will provide them with just that, the accession number. And just run this alignment. So just to like kind of like summarize, right? So far we used BLAST to look for genes that are similar, right? And that's really cool because you can use it, first of all, both in dry lab, both in like exploratory research, but you can also use it when you actually do work in person, like people design primers using BLAST, people working in synthetic biology, for instance, might use BLAST to say like, okay, I have like, you know, this envelope protein for coronavirus 2 for COVID-19, and I want to get like a different organism's version of COVID-19 and run an experiment on that. You can easily use BLAST for something like that. So here we can see the FOXP2 result. What I want to do this time though is click on the alignment. And what's really cool is I can actually like understand more about the similarities between the two proteins this way, right? I can see that they're very similar over here, right? They have the same sequences up top, but what's different is in the human, in the human FOXP2, there's like this region here that is not present in the mouse FOXP2. So maybe that can explain uniquenesses in human speech. You never know. Um, there's also small like point mutations and stuff, but overall, this is really, really cool. And it's something you could easily like just practice on your own because a lot of labs do stuff like this. Um, and it's something that's really like fundamentally similar to what you kind of learn in class. You know that DNA codes your protein. You know that for those of you that are maybe taking biochem, that different amino acids have different properties, right? Some are acidic, some are basic. But this is kind of like something that ties it all together and tells you like a story about, oh, like these are insertion mutations, right? These are a lot of like new amino acids that have been integrated into the sequence. What could that mean for the function of the protein, for the function of the human cells that express this protein, right? So it's something that kind of like is cool for those bio nerds that are in this call. Just moving forward. Um, so like I said, like why bioinformatics, why data analysis, why bother doing exploratory research, right? Mostly it's just that it's really cool and it's a way to practice research in advance. And a lot of it is based on concepts that you're familiar with. You might not know this 100%, right? Like you might not feel comfortable like saying like, oh yes, like blast, this kind of looks intimidating, right? But just practicing learning it, becoming familiar with it is something that's really, really doable. Like for me personally, bioinformatics, like the stuff with Blast, I did it back in like the 10th grade. I didn't know a single person that used bioinformatics. Um, my greatest link to science was like my high school like bio teacher, right? Um, and she didn't know anything about BLAST either. But it's something that's fundamentally simple. It's like a Google search on DNA, and it's something you could just use in terms of like understanding life and something that a lot of labs are starting to take more advantage of. And for those of you that are pre-med, it might be implemented future in the practice of precision medicine. Another thing that's really nice about the way that we have, all of January and, most of December off is since you have free time this break, it's a really, really useful time to just learn skills that you can implement both now and in the future. Like learn how to get acclimated to research, get ready for a lab for the future, or just prepare for your own careers, right? So you can learn coding. So and statistical analyses, if you want to, you can use that to market yourself to labs. I mentioned coding specifically just because, like, for example, what you do in Excel, you can redo that in like R Studio, which is completely free. And it's something that just makes yourself much, much more appealing for labs when you do choose to apply for them to be like, oh, like I'm familiar with R or I'm teaching myself R, right? So it's something that a PI will really, really, really just like appreciate in a student that's trying to apply for a lab position. So to recap, right, what can you do now? We talked about a lot of different things, and there's a lot of content that's been passed around in this presentation. But first of all, you can write a literature review for yourself, right? You can go through papers, practice reading them, and just the practice of doing that is super, super important, super, super useful for the scientific process. The second thing you can do is work on developing your own research skills by running analyses on existing data, right? So exploratory research, like you said. You can compare genes. If you didn't like the idea of bioinformatics, totally okay. I still like you guys. You can still work on analyzing trends in the US population or understanding symptoms that co-vary with COVID-19, right? There are so many options available to you guys, and you don't need fancy software. You don't need $2,000. You don't need anything besides like an internet connection, right? And while it's nice to learn how to code and use software just for like practice, you don't have to, right? You can just practice working with data, and that's the main thing. Like take whatever steps you're comfortable with. And the last thing would be, if you'd like, just try to take an online course. So there's a lot of free courses. Coursera, Codecademy, everyone has courses in bioinformatics, R, and Linux. Linux is really cool for bioinformatics, especially. And yeah, I wanted to just have a bit of a real talk with you guys. Like, I know that the situation is a struggle, right? Like, all of us are going through things like the world right now is so inconceivable. Nobody expected anything like this to happen, right? And I know that this presentation might not have been the presentation that you guys have all wanted, right? A lot of it focused on analyzing data and explorations and like bioinformatics, right? Which might not have been what you necessarily wanted to hear, but it is something that can just help you out in the future. literature reviews in particular are still a strategy that are extremely powerful. And to be honest, one approach that you could take is you could honestly like reach out to a PI, show initiative, and be like, listen, I know that you don't have that much room in your lab, and I completely understand that, but I still want to be involved in research. I still want to, you know, develop skills and, get ready for research when that time comes. Could I maybe do like a literature review, just like to maybe hopefully get into your lab, or can I work on explore like statistical analyses for you just to learn more about your field? And to be honest, like that's something that's really powerful and maybe something that a PI will appreciate, especially if you show that interest and that dedication. Overall, though, the benefits of independent research are firstly, you develop practical skills with real world examples, right? It's not like, you're doing the M&M like experiment, right? Like I feel like a lot of statistics class do that thing where you like roll out the M&Ms and you do like a goodness of fit test to measure like, how many M&Ms there are. Like you don't need to do something like that to practice working with statistics. You can just do something that's more like, relevant to modern day society and to something that's more important in terms of what you value and in terms of what you want to study more, or what you want to research. Not only that, but you demonstrate a lot of initiative and potential. And it's something that professors really, really appreciate. Like when I've applied to labs in the past, and I show them like, oh, like I did this on my own, they really, really, really value that. Like it's so impressive to a researcher, like a professional researcher, to see someone that's trying to emulate what they're doing. And it really just is useful for when you're applying to lab positions in the future, but it's also useful for your own personal development and your own skill development, right? And so we really, really hope that you found this part of the workshop useful. I am so excited to pass things off to Nicolette for our Q&A. So Nicolette, are you here? Just
Hey, yeah, I'm here.
Hi, Nicolette.
That's so funny. I haven't seen that picture in a while.
It's a very nice picture. I like the hat. But
oh yeah, thanks.
Just
yeah, my friend recommended I wear it, and so I wore it, and I was happy about that choice.
It works very, very well. Just to start off, do you mind just like introducing yourself and telling us a little bit about what you do now and how you got involved in research?
Sure. Here, I'm gonna turn on the light quickly because it's a little bit dark in this room. Okay, so it's a little bit better. So I am currently a graduate student in the Ecology and Evolution department at Stony Brook. And so if you look at the slide, you'll see that I study plants and also my bromeliad in the background. So my background is mostly not my Zoom background, but my actual life background, I guess, is mostly in plants. So I always loved science. I never was convinced that I could be doing research. I think I was always curious about like getting to do that kind of thing and being like a hard scientist. I always like looked up to that kind of career, but I didn't totally understand like how academia worked. And I also kind of was like resistant to doing a lot of like certain projects in my undergrad. I still was growing flowers and I was doing a lot of agriculture, but at that time I didn't like link my interests to my academics. And then finally, after a few years, I was like, oh, I'm actually really curious about the way that plants actually have evolved and what the diversity of life on Earth is like. And so I kind of pushed forward and kind of like challenged myself to re-enter into academics and pursue my love of plants in a scientific lens rather than in agriculture. And so now I'm actually a 5th year PhD student, which is funny. In your PhD, time goes by way faster than I think I would have anticipated, but so I'm kind of advanced technically, but I'm still learning a lot. like many of the students here, maybe, I'm not sure, I didn't grow up or have any like exposure to coding or any kind of bioinformatics, but at this point, I also didn't even do it before I started my PhD. I had no idea what it even was when they wanted me to TA for bioinformatics my first semester, and I was like, I don't know what that is. I'm not sure I should do that. But so that is actually a big hurdle for me, but I've actually really enjoyed learning how to do that. Of course, that's not all I do for my PhD. I also grow plants and I take measurements because I'm studying the transitions between annual life history strategy and the perennial life history strategy. So plants that live for 1 year or multiple years. And so I'm looking at genes involved in reproduction and senescence. So ultimately, I'll be using my bioinformatics to look for particular genes and see if they have had any past like selection acting on them and that kind of thing. I think that's pretty much it in terms of a brief overview, but any other questions about me, feel free to pose.
So someone in the class, someone in the chat kind of asked, like, for people that are interested in kind of doing like an independent research project, right? Or people that are really interested in research, do you recommend any specific classes or anything like that?
So that's a good question. I guess the lab-based classes are pretty good in terms of that if you take them seriously. I personally had a hard time connecting to like how structured lab classes are and how little room for like personalization of the questions or whatever that come out of it. But I think like if you are a person that knows you want to do research and you're a curious person, which most of us are, that is what being a person is. So it's like if you take a lab course, what would be really cool, probably, I mean, I can't think of any science or research person that would say no to a student taking initiative and wanting to ask extra questions in that lab class. So I think number one, taking lab classes and getting used to how those things are, like the scientific process from question asking and observation to carrying out research and analysis and writing up the paper is like an excellent framework, but then you can take that hopefully a little bit further and investigate your own kinds of questions, because really ultimately that's the challenge of research, right? Is asking your own questions, putting them all together and being able to take it from like this idea to something that you actually understand and know.
I have a question. So, hi, Nicolette. I know
Hi.
Priya. Hi. I know Priya emphasized or in her slides saying, coding is very beneficial for lab settings. So I was wondering, like, with coding, what type of areas do you recommend us students kind of start with to get a basic understanding? What type of languages have you seen in your research or in your labs? What languages are you most familiar with and you think are most beneficial for students to learn?
So I think R is really good. It's pretty versatile. There's a lot of documentation of what you're doing. And so you can bring they have like lots of different kinds of programs that you can use through R, whereas like other things are not quite as versatile. And so R is like one that's also pushed pretty heavily in a lot of classes, which I think that's the reason that they do that. Because like there's Python, which is a little bit, I mean, I don't actually know much Python, but that would be kind of like a secondary language that a lot of bioinformatics people know. But again, it's a little bit less intuitive for a lot of people. And so R tends to be the thing that people kind of like can jump in on a little bit more quickly. And also in terms of like what I see in my lab setting, that would be, I work with a lot of people that focus on R.
Okay, awesome. Yeah, that helps a lot. I was also wondering, with like from your past experiences, which experience do you think was like the most helpful in producing research with a mentor?
What do you mean producing research with a mentor? Like
working with them, either like developing the research articles or working directly with research, which fundamental experience from your life, looking back, do you think was most important?
That's a good question. So I think I am very like project oriented. So for me, like when I'm working on a project and I can ask questions, but it's mostly like I'm trying to get a task done or I'm trying to find something out in particular. That tends to be the most valuable for me, like when I have an end goal, because that way I'm like, okay, what I'm learning is really cool, but I also have this like thing that's driving me to pass those hard points, you know? Often, so I haven't really had a lot of mentorship. So generally I find that I am asking a lot of different people, depending on the project that I'm working on. So, but that depends on who you're working with and who you're working for, if you can kind of like rely on them to give you constant guidance, or if you have to be a little bit more resourceful. I think it's cool to have to be resourceful, because I think in the end, that's what research is we have to kind of like piece knowledge together. We have to learn from a lot of different people, because no one has all of the knowledge to do one thing, it seems like, because everything's pretty hard and complicated.
Yeah, definitely.
And Nicolette, another
Did that
question.
answer? Mm-hmm.
Sorry, just like another question. So
yeah,
some people mention that, like, you know, if you're trying to get into a lab, like say a company lab, or, you know, I'm just going to adjust the question a little bit, Richie, if that's okay, and just like either a company lab or a lab in general, right? And you worked on literature review or exploratory research in your free time. Like if you were like taking in an undergrad student, right? Like how would you like them to market that to you in your application? Like, is it something they would put on the resume? Like, do you want them to talk to you about it? Do you want them to bring like a literature review, like if you were taking in an undergraduate student or something like that?
Okay, so generally I don't ask for a CV for my undergrads, but I guess that probably varies. But if someone wrote a literature review and they thought it was like, you know, relevant to my work, then it would definitely be cool to read it. And I guess if it's published, that would be something that would be good on the CV, but I'm not sure it would necessarily belong on there, just if you wrote it, you know. But it's definitely something that would be cool to like see what your interests are and see the types of connections that you're making. So writing them and sharing that with a person, that sounds awesome. Like sharing that with a potential mentor is like, that sounds beautiful. I've never had anyone come to me with something prepared, you know. I just have people come to me saying, oh, I have this interest and I hear you do this, so I was wondering if I could help you in some way. Generally, that's an awesome thing for a graduate student, but I guess it always depends on the project.
The question, just kind of going off of what Priya was asking. So, what is your perspective on this type of independent research and like these types of projects?
So I actually hadn't like thought about this as a concept before this kind of the discussion about this meeting. So I think it's actually a really wonderful idea. So in parallel with my research, I'm also trying to cultivate as through artwork people to be more interested in pursuing science in kind of like this more individual way. So trying to figure out mechanisms to get people or young people to ask questions and investigate them on their own, because what is more fun to like work really hard on other than something that you care about? You know, it's like, it's a beautiful thing. So I think that the idea of doing independent research and fleshing out your own ideas and taking it from like just like idea formation to this like actual piece of science is a really, really cool thing. And I think that it would not be a waste of time for anybody to go through that process, even if they're not a scientist. I think that like developing your own ideas in that way is, you know, a huge thing for anybody who's learning.
Yeah, I agree. Thanks for the insight.
Sure.
Nicolette, so someone asked in the chat, what is something surprising or interesting you found in your research?
Like in terms of the outcomes of the data, or like in terms of the process of doing the research?
Let's go with both for now.
Okay, cool. I guess. So right now, I haven't had like any like cool findings. I guess when I grow my plants, so I go out into the mountains, the Rocky Mountains, and I collect little seeds for these little plants that I grow. They're called Drava. It's a genus. It's very diverse. There's a lot of little, they're like the tiniest little plants, and they grow on mountaintops and in meadows. And so when I collect them, I don't totally know what they are, and because it's just their seeds and I'm just guessing all the time. And so when I actually grow them, I get to see how they grow and how they grow differently in the growth chamber. I guess that's what always is surprising to me, because different populations. So it's like I can collect the same species in a bunch of different places, like throughout Utah, for example. And the exact same species will have very different characteristics depending on where they were collected. And sometimes they're like, in terms of like my research, it's this annual and perennial thing. So I have some species that are perennial when you grow them in the this like kind of like standardized environment that I have them in the growth chambers at Stony Brook. And then some of them they don't die, or they grow like some weird, they grow some weird leaves like at this top. That is what keeps them alive for multiple years. So it's that for me, since I'm a plant person, I guess that would be the most exciting thing, is like actually experimenting with my plants and seeing what they do under my growth conditions, and like looking at that. And then in terms of the process, I guess bioinformatics is kind of like a constant form of discovery for me, because there's so many different ways that you can put stuff together. And like when you read these manuals, it seems like there's like one way to do something. But for me, the fun part is like you pick up one little piece from one documentation, and then you pick up another piece from some kind of like Slack conversation, and then you get another thing from like BioStars or something. And then the fun part is always like when you put that together and you just kind of like experiment, then finally it's like, oh wow, like those things worked. And it's cool to see, like, I don't know, I guess that for me is like a fun process of seeing what's interesting and how you can just kind of like put anything together as long as you think it through. Again, it's like this, what's the valuable part of these independent research things is like you have to think it all through. You have to put together the puzzle pieces. And in bioinformatics, to me, it's like cool whenever the puzzle comes together. Oh, cool. Yay for plants.
Knowing what you know now and like your place as a grad student, what advice do you have for people that are looking to get into research or even like freshman students in college?
I think, so I'll be honest with you. I think that what the most important thing is that you just follow whatever drives you. So whatever it is that makes you curious, whatever it is that keeps you wondering about it, like then you can't help but think about it is what you should follow. So especially if it's research, it's like you want to understand something, right? Like that's what research is. You want to make new discovery, you want to think something, you want to think about something in your own way, and you want that to come out into scientific papers or whatever. And so the biggest thing that you have to develop is this like kind of like personal drive to create that unique science. So as long as you kind of attach yourself to that thing that you love, like, so for me, it's plants. I don't know. Everybody has, you know, lots of people like lizards, people like trees, or people like spiders, for example. You know, it's like whatever it is that you're like curious about, just like dig into it and like love it, because that's what will drive you. That's what will bring you through like the hard parts of what research is. And of course, like in the more academic part is like, don't be afraid to get yourself involved into things that are making you interested. I know that's what, I guess that would be one thing that maybe I would regret. I mean, I spent time doing other stuff, you know, I like was always growing plants. I was always experimenting in that way, but I was always a little bit afraid to follow the science part. And I think it would have been cool if I wasn't afraid to do that earlier in life. Because I'm a person, I struggle with reading. I'm dyslexic. I have like all this stuff. So I was always like, well, I can't really do that. So, but just not being afraid to do whatever you want is a really lovely thing.
And that was beautiful, Nicolette. I'll just say that was like super beautiful.
Thanks.
Another person was asking in the chat, just like, how is a literature review different from a research paper? I guess more of a technical question, but still very important.
Yeah, that's it. Totally. So a research paper is like posing one scientific question, and it's generally something that you've come up with. So you've had the scientific question, and then you come up with a procedure that will get you to answer that scientific question. And so the research paper just walks through that process. So it states like the question and why you're trying to figure it out, and then you walk through with the methods what you've done, and then you report what you've done, and then you talk about like the relevance of it and why it's important on the grand scheme of things. But a literature review would be something that you're like broadly interested in, and you know that there's a lot of like research that's done about it, but as research happens, it's like it's always expanding, so we're always finding out new things, and sometimes they're expanding in parallel. So there's related ideas that are developing over time, but someone needs to make those connections to get a better picture of like that idea in general. So like, you know, people do literature reviews about certain genomics things like transposable elements, and that is an expanding field all the time. People are obsessed with transposable elements because, you know, they contribute to genome size and they contribute to lots of duplications. And so people are interested in how they contribute to evolution, but they find out one thing about one kind of TE over here, and then they find out, you know, about another TE, and, you know, not everybody is in communication. So it's really helpful when these people that are doing literature reviews kind of like put that information together into a story so that people can know where the field is right now in this type of this one topic. and so in terms of like format, research papers tend to be pretty like structured, you know, methods, introduction, methods, results, conclusions. And then, but a literature review actually tends to take a little bit more free form. There's always like a pretty like, there's like an introduction that tends to be pretty similar to a research paper, but then the way that it goes is, you know, depends on what's been happening in the field. Generally, there's like different sections that people outline. So if there's like some major themes that are newly developing, there would just be multiple sections that are discussed in the paper.
Awesome. And we can just take like one last question from Nicolette. If anyone has like one final question.
I had like two questions, but I guess I'll ask one. So, I was wondering, like, with public data approaches and like the public tools, I know Priya was mentioning Blast. What types of like public tools have you seen the most, and what do you recommend for beginners that don't have any experience in like information technology and stuff?
So one thing, in addition, because I think you were on NCBI, right, and talking about the different sequences that are available there. And then, so there's another thing. So one thing that a lot of people are doing now because of COVID is like using data that's available online, just because like there's so much less lab work that can be done, so less actual DNA can be obtained. And so people are going on to, you know, places where data is deposited and using the information or the, you know, the sequence data or the whatever. They generally it's all available. So there would be the raw sequences, and then there would be assemblies that are available and things like that. So that's another thing. And so generally from a scientific paper that has done the work already, where that's where you would find out where it's put up. So at the bottom, generally there's like a statement of data availability, which people, I think it's generally required that people make those data available, mostly for that transparency of science reason. But then also there's just this idea that everyone is, you know, sharing science. So that would be where they post like a link to where the actual sequences are stored. And so you download them and you can work with whatever they have there. So whatever research papers you're interested in, generally you can use them and develop your own questions with their actual data.
Okay,
And I know I said,
awesome.
like,
Thank you for
final-
answering.
Sorry for cutting you off, Misha. But one final question, actually, we'll make this like the final question. But Cuomo just asked, like, what inspires you, Nicolette?
Okay. I guess always that's a multi-dimensional question, but I'll try to be concise. So my inspirations lately are mostly in the human connection to the natural world. Probably driven by the fact that the way that our society has developed over time, we've kind of like lost the connection to what nature is and how many crazy things there are for us to understand and look at, you know, like even if you're just standing out. So I'm a person who just like stands on the sidewalk and will like hang out with whatever like weed is there and like watch the like millions of aphids that are like on it, you know. And I'm like, oh my gosh, there is this plant insect interaction that is happening right here and nobody cares. And I'm like, there's so much we can learn. There's so much that you can teach someone just from like showing this like one plant that's covered in aphids, you know? And so for me, what keeps me like in science is kind of like following that idea for myself. So trying to stay connected to like, you know, what those cool like things are if you just kind of like hang out with nature and just watch it, you know, because it's unfolding right in front of you. And so it's easy to lose track of that as a graduate student, but I do my best to hold on because that is what keeps me going. And then also I'm also like kind of deeply inspired by young people. So it's really joyful for me to be here. I'm inspired to like try to keep people interested in nature and try to maybe hopefully have future generations break the cycle that we're currently in and use that curiosity that we all have to try to find new solutions for what our future will hold. And I think that that is in science, you know, it's like in our understanding of the world around us, because the world around us is always changing. And right now it feels like it's changing for the worse. So we need to kind of like figure it out. And that just comes from us interacting with nature. So I guess, you know, that's what keeps me going every day.
Thank you so much, Nicolette. I hope that a lot of the people that are here were able to take a lot out of it. I took a lot out of it still. So I hope that, you know, all of you kind of like find semblance in the fact that like, yes, everything is kind of hard right now, right? But we are kind of going through it, and there's things you can do if you want to explore your passion. There's still beauty in the world that you can explore. We just wanted to give like a couple reminders at the end here. Firstly, like for those of you that are like exploring your interests, right? One really cool thing that's up and happening event is the SBYIR Symposium. So we are actually hosting this event where a lot of undergrads are actually like presenting their research, and we're also having a keynote speaker coming from Stanford University, and he'll be talking about neural activity and the effect of that on visual perception. Really cool at 1 p.m. And it's really awesome because a lot of the work that people are going to be presenting is largely computational. So you can kind of see the connections. I don't know for sure if anyone's talking about BLAST explicitly, but a lot of the work that people are going to be presenting on are stuff that they've been working from their computers from home, even like people are analyzing the stars using software. Like all of this is really cool stuff. And kind of like as incentive, if you stay for 2 hours, you'll get not only a sticker, like a science sticker, not like anything special, but like a little sticker, and also a $25, a chance to win a $25 Amazon gift card. So it's like, it's nice. It's money. Money is always nice. If any of you have friends that might have been presenting, this is the full schedule of presenters and the fields that they're in. And this should also be on the SB Engaged event, but event page. But essentially, like if you're interested in exploring your interests, you don't have to come for the whole time. You can just come for one presentation that interests you. But there's a lot that you can learn about. A lot of the students that are going to be presenting are going to be talking about the research that they do on campus, right? So it's also a way for you to learn about what's going on with the labs at Stony. So it's like a really cool event for those of you that are interested in research. And what's nice about this event is that it's kind of like feedback oriented. So these students are going to give a 15-minute presentation. There'll be like a 5-minute Q&A, but then afterwards, there's going to be like 5-minute feedback from the Alan Alder for Communicating Science. Like they're going to be explaining like how to present research and give feedback on presentation skills in a way that's like focused on communicating science. How do you educate a community and explain your research well? So if any of that sounded cool to you, definitely come through. I think a couple of the people that are presenting are also in this call, which is really awesome. And so we have a Kahoot prepared for you guys that love Kahoot. So I will get that going for you guys. This is my first time running a Kahoot, so I'm sorry if this doesn't work the way that it should. I will host a Kahoot. And are you ready to play it? Awesome. Okay, so if you want to play, you can just go on. I know this. Yeah, just like Kahoot.it. And you can just click on it. stop the recording.